Guys, in the last video lecture we understood, okay, we understood about the apoptosis, okay, from the intrinsic pathway, about the intrinsic pathway. So we had seen this thing, okay, we had seen the video, we understood here what is happening, we summarized. Then yeah, this we have already discussed in the earlier video. So in this video what we will be discussing is the extrinsic pathway. So the last video was intrinsic. So intrinsic was from where? Intrinsic was from the within the cell. Within the cell from the where? From where? From mitochondria. How? Because of the permeability of the mitochondria is increased. Permeable to what? Permeable to cytochrome C. What it does? It goes and binds to the caspases. And caspases and mitochondrial cytochrome C, they combine together, they form some kind of Sudarshan chakra, and they quickly cleave the cell from everywhere inside the cell, cytoplasm, then the organelles, cytoskeleton, everything. Okay, so in this video we will be talking about extrinsic death receptor initiated pathway. So in this what happens is, so there is something else coming from outside and initiating the apoptotic pathway in the cell. तो बाहर से कोई आ रहा है, सेल के अंदर apoptotic इंड्यूस कर रहा है. कैसे हो रहा है, क्या हो रहा है, लेट्स सी. सो वेयर इज माय पॉइंटर, laser. So the extrinsic death receptor initiated pathway, this pathway is initiated by the engagement of the plasma membrane death receptor. ठीक है, engagement of the plasma membrane death receptor. तो plasma membrane में कुछ death receptor होता है. ठीक है, कौन सा receptor? देखो, several. There is like thousands and millions of receptors in every cell. Out of those thousands and millions, there is one kind of receptors which is called as death receptor, which is responsible for the death of the cell. So there is death receptor are members of the tumor necrosis factor. So these are all family, okay, receptor family. So tumor TNF, this will you will come across this TNF everywhere in the pathology, here and there, inflammation, AO, okay. So receptor family that contains a cytoplasmic domain. Okay, so see here, this is my plasma membrane, this is my cytoplasm, and here is my death receptor. So there is one component which is outside the membrane and one component which is inside the membrane. The membrane, the component which is inside the membrane, this is called as the death domain. Okay, this is called as death domain, and the one which is outside the cell membrane is just a receptor, the death receptor. The outside one is death receptor, inside one is death domain. This one is called as FAS, this one is called as FADD, or whatever it is. FADD is fast associated death domain. So FAS and this. So what's happening here? As you can see in the picture is very clearly saying that something is coming from outside. It is forming something called as FASL. What is FASL? This is nothing but the FAS ligand. So we need a ligand which is attaching on the receptor, and then there is some signals going across these receptors, and there is formation of the FADD, procaspase 8, and I have highlighted this point many times in the previous lecture. If you have not seen, you have to go and check that. So in that lecture I have told you that caspase 9 was for the intrinsic pathway, caspase 8 is for the extrinsic pathway. So as you can see here, so we'll read. This death domain is essential for delivering apoptotic signals. Some tumor necrosis factor receptor family members do not contain cytoplasmic death domains. Their function is to activate inflammatory cascade, and their role is triggering apoptosis in much less established. Okay, what? So there are certain cells which do not have this death domain. Instead, they have inflammatory cascade. So there is inflammation. Okay, so maybe there is an inflammation, and this inflammation is a whole new topic. Okay, it might take 1 hour lecture or something. Inflammation, that is different topic. There is a 10 marker in Mbbs, so that is a different topic. Anyways, we will discuss. So the best known death receptor are the type 1 tumor necrosis factor, which is also called as TNFR1, and a related protein called as FAS. So as you can see here, this is FAS, which is CD95. This is also called as CD95. What is CD? Cluster of differentiation. And this is present where? This is present on the immune cells. Okay, all the WBCs, they have CD cells. Okay, T cells as different, B cells as different. So you will come across this many times. So CD, you just remember CD95. 95 is for the death receptor. Okay, but several other have been described. The mechanism of apoptosis induced by these death receptors is well illustrated here. A death receptor expressed on many cell type, the ligand for FAS is called as FAS ligand. So यहाँ पे देखो, this FAS is nothing but the ligand. Okay, so the ligand is coming and attaching to the receptor. The FAS cell is expressed on the T cell. This is important. So as I have mentioned several times, something is coming from outside. Something is coming from outside and then it is initiating the signals or whatever. So that something is whom? This is nothing but the T cell. The T cell or the cytotoxic cells, okay, these cells, they come from the outside. They take care of these cells. Now when is it important? So let's suppose ki let's suppose there is a cell, inside the cell there is invasion of the virus or something. Now that virus has entered inside the cell and virus, that cell has to be removed, okay, but the cell is not able to initiate its intrinsic apoptotic pathway from the mitochondria. Something virus has done inside the cell, due to which the cell is not able to undergo self-apoptosis. So at this moment the body has to do something. So he sends whom? He sends the mighty T cells. So the T cells goes there over the cell, they see the cell, okay, so they scan the cell, which cell has been invaded by the virus. So they scan them, and when they identify okay, this cell has been invaded by the virus, so they just go with their FAS ligand and they go and attach their FAS ligand with the death receptor of that cell, and then there is initiation of all this triggering cascade effect, and then there is procaspase 8 and all, and then there is apoptosis. So the cell and the virus, both of them die. Okay, so we have successfully taken care of the infected cell. So same thing is trying to say here, the ligand is called FAS ligand and they have expressed on the several cell. Okay, so one more example is, let's suppose there are formation of the white blood cells which are able to recognize the self antigen. What does it mean? It means this cell, the WBCs which were made, their only job was to protect us from the outside invaders, the foreign invaders. They were supposed to kill the outsiders, but instead these self-recognizing antigens, they are killing our own cells. They are not killing outside cells, but they are killing our own cells, which is pathologic. So we need to clear these kind of cells. Okay, so that example is trying to give here ki we need to clear this kind of cells because they are not dying on their own. Okay, they are not dying on their own, so they have to be taken care from the outside mechanism, and so the body is sending the T cells to take care of these kind of faulty cells. Okay, so on some CTL, what is CTL? Cytotoxic T lymphocytes, that virus killed infected and tumor cells. Also, as I have already discussed about the virus and also the tumor cells, so there is extensive proliferation of some abnormal cells, so they are also abnormal, so body recognizes it, the body identifies it. Okay, it goes and quickly kill all these tumor cells. Okay, but if the tumor cells are stronger enough, then gone case. So when FAS ligand binds to FAS, 3 or more molecules of FAS are brought together and the cytoplasmic domains forms a binding site for an adapter protein called as FADD. Here you can see FADD. What is that? FAS associated death domain. So once this FAS, this is one death domain, this is the outside component which is the FAS, okay, protein, the inside one is death domain. So this is one. When this FAS ligand is coming, many of these several proteins, they are coming together. Okay, after they come together, then there is formation of the FADD, which is FAS associated death domain. Once the attachment of this domain is done, then there is initiator of the procaspase 8. Remember again, I am telling 8. Okay, there we had caspase 9. So bringing together multiple caspases molecules and leading to autolytic cleavage and generation of the active caspase 8 in turning active caspase 8 inside the specific executioner. Okay, so at the end there is apoptosis. You know that we had discussed already in the previous lecture. Go and watch that. Okay, if you are not watched. Then this is something interesting. Sometimes what happens is, let's suppose some virus, okay, and he is very intelligent. So these intelligent viruses, they enter the cell and they do something inside the cell that the cell is not able to initiate the intrinsic pathway of apoptosis, neither it is able to neither the body is able to kill those cells from the outside or extrinsic pathway. How is that? That is, there is something called as flip protein. So this extrinsic apoptosis pathway can be inhibited by a protein called as flip, which binds to the caspase 8, thereby blocking the FADD. So this FADD, if we can block here, so once the virus they enter inside and then they block this thing, and if they block this path of the pathway of the apoptosis, then this won't be initiated, and if this is not initiated, then there is no apoptosis, right? So virus is so intelligent enough to come and block this step, and that is through some protein is he's calling them as flip protein. Okay, so which binds to caspase 8, thereby blocking FADD binding but cannot activate the caspase. Some viruses and normal cell produce flip as a mechanism to protect themselves from the fast mediated apoptosis. So yeah, that was it. The extrinsic and intrinsic pathway of the apoptosis are initiated fundamentally different. So yeah, intrinsic pathway was from the intrinsic within the mitochondria, the release of the cytochrome C, caspase 9 and everything. Okay, and then in the this thing, extrinsic pathway, something else is coming from outside. Who is that? The T cell. What does he have? He has fast ligand, and what this cell has? Fast receptor, the death receptor. Okay. So for instance, in hepatocytes and pancreatic beta cells, caspase 8 produced by the fast signaling cleaves and activate the pH3 only protein, which is BID. So I have already discussed this in the previous lecture. BH3 only protein. This is the domain in the BCL family protein, the apoptotic protein, which has the ability to induce or trigger the apoptotic pathway. So which then feeds into the mitochondrial pathway. The combined activation of the both pathway delivers a fatal blow to the cell. Okay. So an inhibitor of the DNase, making the DNase enzymatically active and allowing DNA degradation to. Okay. So at the end, he's just saying ki, so once the apoptosis is being initiated, everything is just getting cleaved and destroyed, and here and there everything is forming blobs, and then there is loss of cell. Okay, and at the end the removal of the dead cell, the kachra sets ka department. Okay, who are the kachra sets? Macrophages.
150 रुपया देगा.
Okay, so the formation of the apoptotic bodies breaks the cell up into bite-sized fragments. So as I have already told in the first lecture, also very first lecture, why is the formation of the blobs? Because it is very easy for the body or very easy for the macrophages to clear up. So you imagine if I give you this big of food, okay, and if I tell you to put whole thing inside your mouth, will you be able to? No. But if I cut down the big components of the food into several small like your mother used to do when you were a child, so at that time that was very efficient to eat, right? So the formation of apoptotic bodies breaks up into bite-sized fragments that are edible for the phagocytes. So apoptotic cells and their fragments also undergo several changes in their membranes and actively promote their phagocytosis. So they are most often cleared before they lose membrane integrity and release their cell contents in healthy cells. Phosphatidylserine is present on the inner leaflet of the plasma membrane, but in apoptotic cells this phospholipids flips out and is expressed on the outer layer membrane. So in the previous lecture also, as I told, eat me, find me and eat me signals was released by whom? Was released by the apoptotic, the cell which is undergoing apoptosis. Why is that? So that the macrophages, the kachra cells, they come in and they clear up as quickly as possible, so that the body is not undergoing any kind of inflammation, which was really good mechanism. So how is that process being performed here? So in the plasma membrane there is some proteins. Okay, so the phosphatidylserine is present in this phosphatidylserine that is initially present inside the cytoplasm, inside below the leaflet, but in case of apoptosis it flips, it comes outside, it is expressed outside, so that this phosphatidylserine is being this is like chara. So the macrophages, they can find this phosphatidylserine, so that they can find them and they quickly come and they understand, okay, this cell is undergoing apoptosis, so let me just wait, let the apoptosis happens, let the formation of the blebs and everything. So once it breaks down into its components, they quickly engulf it. Okay. So other membranes, and there is recognized by several macrophage receptors. Cells that are dying by apoptosis also secrete soluble factors that recruit phagocytosis. The macrophages themselves may produce proteins that binds to the apoptotic cells but not live cells, leading to their engulfment. So it is very understandable what he is trying to say. If not the apoptotic cells, the macrophage itself is releasing certain chemicals which is going and attaching on those cells which are undergoing apoptosis and not the healthy cells, so that there is a differentiation between the healthy and the damaged cell or whatever, so that it can go quickly and clear the debris. So that is the eat me and find me signals, and this process is called as epherocytosis. What? Epherocytosis. This process of the apoptotic cells phagocytosis is called epherocytosis. It is so efficient that the dead cell disappear within minutes without leaving a trace, and this is very much important to control the inflammation. In addition, production of the pro-inflammatory cytokines is reduced in macrophages and have ingested apoptotic cells. Together with rapid clearance, this limit inflammatory reaction. Are we done? Done. Okay. So this was about the extrinsic pathway. So 3 videos have been done. I also have my exams, so I have to read for them also. Bye bye.